think I came to Cambridge almost at the right time, because when I came, this was in the early 70s, this was the time when there was a lot of pioneering research that was going on in Cambridge on mammalian development. So this was a hugely exciting period, and the whole environment was highly conducive to thinking in a fresh and in a new way. There was a lot of freedom to do curiosity-driven research, and we stumbled on something which turned out to be quite an unpredicted and a profound discovery. So I was born in a small town in Kenya near Lake Victoria. My parents were not very restrictive, so they allowed me to wander around, and so I would go around looking at all of these small creatures that I would see. It was a very kind of free and happy time. My parents hadn't gone to university, and none of the people I met during my time growing up had been to university for me. I didn't have the idea that you could have a career in science. But I started to get interested in how there could be such biological diversity in the butterflies and fish that I was seeing around me. Actually, I was slightly lost before I got the offer of doing a PhD with Bob Edwards, who was pioneered the in vitro fertilization for which he received the Nobel Prize. Bob generously gave me the opportunity working on a question that I was excited about. So I decided to work on Parthenogenesis, which is virgin birth. There are some vertebrates where, like lizards, fish and frogs, that can develop parthenogenetically without any contribution from sperm. And so the question was, can this happen in mammals? Can it happen in us? So we started working on this and did lots of experiments, and none of them worked. But this actually was the prelude to what I think was a really big breakthrough in the understanding of mammalian development. The most profound part of the discovery was that the genes that are inherited from mother and father are virtually identical genetically. And so the prediction should be that it should not matter where the genes come from. The parental origin should not matter. But what we found was that there's a difference, functional differences between the genes coming from mother and father. So those genes that come from the mother are important for the development of the embryo, and those that come from the father are important for the development of the placenta. And both of them are necessary for full-term development of mammalian embryos. I call this phenomenon genomic imprinting because what it told us was that the parental genomes at fertilization come in with a memory of the inheritance. They remember whether they came from the father or the mother. And so that became an important part of understanding the mechanism for embryonic development later on. This is quite a remarkable discovery and completely unexpected. And so we needed to find out more about what was going on. And it became clear that the whole process of imprinting, that is the introduction of these marks, was occurring in the germ cells, which are the cells which give rise to sperm and eggs. So we started to look for genes that were functioning differently depending on their parental origin. And indeed, we found some genes that were either on or off, depending on whether they came from the mother or the father. And when we examined these genes, the location of these genes, we in fact did find that there were these tags, this kind of what we now call epigenetic tags or DNA methylation tags, which doesn't alter the underlying DNA sequence, that were put on during development of sperm and eggs that were transmitted at the time of fertilization to the embryo. There's only about 1% of the genome that is imprinted in this way. So there are about 200 genes approximately that are imprinted, but they have very profound effect on mammalian development. There is a cost to this phenomenon because there are about 14 human diseases, including cancers, which occur because of faults in the imprinting process. The implications of what started as a curiosity-driven research and a very bizarre phenomenon of genomic imprinting is hugely important in many diverse ways. And so, although it started up with a single question, the field is now huge and gone in many different directions.